Hello students, I hope you are doing well. So in this new chapter, we will be learning about nucleic acids, different nucleic acids found in DNA and RNA, and structural components of nucleotides, and difference between DNA and RNA, and lastly, DNA replication and DNA transcription process in details. So the components of the nucleotide. So there are basically three components of a nucleotide. Basically the phosphate group, this is the first component. The second component is the sugar, that is a five-membered sugar, that is also called ribose sugar, five-membered, or pentose sugar. Actually five-membered is called pentose sugar, and in case of DNA RNA that is ribose, in case of RNA that is ribose, and in case of DNA that sugar is deoxyribose. That means one oxygen is less in the DNA sugar. The third component is a base, the nitrogenous base. So there's mainly two type of base. One is purine base, which are double ring. You can see this is double ring, and these rings are heterocyclic. And the other type of base is pyrimidine, that is the single ring base. And this base is also heterocyclic. And this purine A and G is found in both DNA and RNAs, but in case of this thymine is only found in DNA, the DNA base. And this uracil is not found in DNA, but this is uniquely found in RNA. But the cytosine is found in all this cytosine is found in both DNA and RNAs. So note that when these three components are together, then they are only called a nucleotide. But when these two components are together, this base and sugar are together, they are called nucleoside. But at least one phosphate group needs to be attached to nucleoside in order to call it nucleotide. One difference between this ribose and deoxyribose, you can see this ribose sugar, the 2' carbon is having a hydroxyl group OH, but in case of this sugar, that is deoxyribose, that means this oxygen is absent here in the same place, the 2'. So that's the main basic difference between the sugars of DNA and RNA. Is that deoxy, the 2' carbon has one less oxygen. Actually the whole sugar has one less oxygen, but that less oxygen is in the 2' position. So that's the difference of the sugar or ribose or deoxyribose. So moving to the next slide. So these are the DNA nucleotides. I mentioned that the nucleotides need to be three components, right? You can see this is phosphate group, phosphate is attached to the deoxyribose. And this is DNA, so this is deoxyribose sugar, and this is thymidine, and together they call deoxythymidine monophosphate. Monophosphate is because actually this is nucleotide, but the full name is, you can say, deoxyribose monophosphate, deoxythymidine monophosphate, but together they are nucleotide. And this base is thymidine and this sugar is deoxyribose, and this phosphate group is attached to the 5' end, and we will see that how this nucleotides are joined to form the DNA strand. Actually the other phosphate group will join in this position, this 3' hydroxyl group. We will see that. So these four are the DNA nucleotides. You can see there's no uracil here. This thymidine is present because there's no uracil is present in the DNA. But in case of RNA, if those are RNA nucleotides, we will be seeing that this thymidine is replaced with uridine. So this one will be uridine or uracil. So these are the DNA nucleotides. These are mainly joined together by a phosphodiester bond. We'll see that. And also note that the numbering starts from this nitrogen position, this base position, this 1, 2, 3, 4, 5, 6, and then the numbering starts from this ribose sugar. So as there is already 1, 2, 3, 4 numbering is present, so we cannot number the sugar 1, 2, 3. Rather we do 1 prime, 2 prime, 3 prime, 4 prime. So this prime position actually denotes to the sugar carbon of the sugar molecules. But this 1, 2, 3, 4 is the actually numbering of the nitrogen and carbon of the base, nitrogen base. So also note that the position that is important for the sugar is the 3 prime end carbon and 5 prime carbon. These two carbon are important in case of the linking or joining of the nucleotides. And other position is the 2 prime, because the ribose and deoxyribose mainly differs in this position, this 2 prime, because of the one less oxygen of deoxyribose sugar. So that's the main differences. So going to the next slide. So depending on the sugar and the base, we call it ribose or deoxyribose. So this is one basic unit of a nucleotide, and basically these nucleotides are overly repeated through the phosphodiester bond and form the DNA polymer or the RNA polymer or the strand. You see, so repeating unit of deoxyribonucleic acid if that is DNA, and the repeating unit of ribonucleic acid if that is RNA. So we will see that how those nucleotides are joined together. And also note that is a typical structure of adenosine ATP molecule, adenosine triphosphate. So I mentioned that when they are together, like one phosphate group is attached, then we can call it nucleotide, but at least one. But if there is three phosphate groups, the triphosphates, we also call it nucleotide. So actually when they are free in the cytoplasm, the nucleotides are free. Actually they are in this form, this triphosphate form. But when they are incorporated in the DNA strand or RNA strand by the DNA polymerase or RNA polymerase, then these two phosphate groups are cleaved or cut off, and the energy is released, and those energy is mainly used by the DNA polymerase to incorporate this nucleotide into the growing strand of the DNA. So don't think much now, we will see those in the DNA transcription DNA replication process. But note that when at least one of the phosphates are attached to the ribosome base, they are called nucleotide, or in other words adenosine monophosphate. But when there is no phosphate, they are called nucleoside. You can see this from this portion to this portion, these two are joined without a phosphate group, they are called the nucleoside. If that is the adenine, the base adenine, this is the adenosine or adenosine. So moving to the next slide. So the DNA and RNA, the difference, the main difference we know is the structural difference is, the first structural difference is DNA is the double strand, the RNA is single strand, and another structural difference is the sugar structure. We know that in DNA that is deoxy, that means one oxygen is less in this 2' position, but in case of RNA this oxygen or hydrogen or hydroxyl group is present. That means there is a ribose. That means the full hydroxyl group is present, but in case of DNA there is no oxygen, only hydrogen. Also another difference is the base. The thymine is uniquely present in DNA and uracil is uniquely present in RNA. So these are the main basic structural differences of DNA and RNA, but in case of functional differences, the function of DNA, like storing genetic material, like giving the cell instruction to make the protein, so these are the functional characteristic functional difference between DNA and RNA. The RNA mainly functions as a messenger between DNA and protein. And the RNA, like non-coding RNAs, have different functions. We know that. So those are mainly the functional differences. There could be a lot of functional differences, but the structural differences are limited. So in the left side, this we see a strand of DNA. There's just one strand is shown, but in case for simplicity, just one strand. But in reality, there's two strands of DNA are joined together. So you can see, if you can see this, these four bases are shown here, like thymine, adenine, cytosine, guanine, and all of the nucleotides are joined together. If like you can see the middle is the phosphate group, and how those nucleotides are joined. Those nucleotides are joined by this oxygen and this oxygen. So this and bonding between oxygen is called ester bonds, right? So in one phosphate group, you can see this one phosphate group is linking two sugar molecules, this one and this one, sugar molecules of the nucleotide. So that means this phosphate group is linking the nucleotides, and this linking or the bonding of these two oxygen is called the phosphodiesterase bond, or phosphodiesterase link, or phosphodiesterase bond. Also note that one ester bond is linking this 3' end of one sugar, and the other one is linking the 5' end, like 5' carbon, and the other one is there was the 3' hydroxyl group, right? So this is mainly important, like linking from the 3' end of one sugar of one nucleotide and linking into another sugar into that 5' end of that sugar nucleotide. So in the right side there's the same thing, but it is for the RNA. So you can see the base is uracil. There is not thymine anymore, and adenine, cytosine, guanine. Four different bases of RNA, and here the four different bases of DNA. But in case of DNA, just one strand is shown, but in reality there should be two strands. So these are the many differences of DNA and RNA and how those are formed in strand, like through the phosphodiester bond.